Welcome to the Deep Dive. We're here to cut through the noise and give you clear insights from key research. Today, we're tackling something huge, the intersection of two massive global environmental problems. We're talking plastic pollution and biological invasions.
Yeah, it's fascinating, really. You've got plastic, this physical, chemical contaminant everywhere, and then you have non-native species, these biological disruptors changing ecosystems. For the longest time, science treated them as completely separate issues.
Like two different worlds.
Exactly. Which, as we're finding out, was a major oversight because they're now constantly bumping into each other systematically.
So we're diving into a brand new systematic review that tries to map this out. It's called Plastic Pollution Meets Biological Invasions, a systematic review of emerging interactions. The lead authors are Sousa, Ronaldo Sousa, and Padilha. It's a 2025 preprint, so it's really fresh off the press.
Right. And what they did was synthesize 146 peer-reviewed articles. It's basically the first global look at this whole emerging field.
Giving us a map, essentially.
A first map, yeah. And the scale here is just, well, staggering. Think about plastic production. It doubled in less than 20 years. We went from about 234 million metric tons in 2000 to 460 million in 2019.
Wow.
Now, picture all that extra plastic out there floating around, sinking, breaking down. It's acting like a new kind of mobile habitat, a synthetic one. And that fundamentally changes the game for how species move around, you know? It breaks down these natural barriers that took, like, millennia to form.
Okay, let's unpack this then. The sheer amount of plastic is essentially becoming a vector for life, but maybe not the life we want spreading. So the review's mission was to figure out the specific ways this collision is happening in the real world.
Precisely. And I think the paper's biggest contribution is identifying these three main ways, these recurrent pathways, where plastics and non-native species actually intersect. It cuts through a lot of scattered studies.
Okay, let's get into pathway one.
Yeah.
This feels like the most obvious one, the physical link. You called it the rafting effect?
That's it, pathway one. Dispersal and colonization. So think of larger pieces of plastic, macroplastics, especially stuff that floats. They act like little rafts. They provide a hard surface, a substrate for organisms like algae, invertebrates, even microbes to attach to. And then these plastic rafts just drift across oceans, lakes, rivers, carrying these non-native species to totally new places where they might take hold.
We hear about the Great Pacific Garbage Patch, right? But this framing makes it sound less like just trash and more like mobile artificial reefs carrying potential invaders.
That's a good way to think about it. And the review found some really concrete examples.
Like what? Anything particularly surprising?
Well, they mentioned non-native Tubostrea corals found growing on floating plastic debris off Brazil's coast. That directly shows plastics helping species expand their range in the ocean.
Okay, that makes sense for marine systems.
But here's one that really jumps out. They also found non-native barnacles attached to plastic leg bands. You know, the rings put on birds for tracking.
Wait, on bird leg bands? You mean plastic is helping things hitch a ride through the air via birds?
That's what the evidence suggests. It's indirect, obviously. The bird carries the plastic ring, the barnacle is on the ring, but it shows this potential for long-distance aerial transport.
Wow.
Connecting ecosystems that you'd never think would be linked that way. It's not just ocean currents and ship hulls anymore. The pathways are getting, well, weirder.
That is genuinely surprising. Okay, so pathway one is physical movement, the rafting. What's pathway two? You mentioned altered environments.
Right. Pathway two is altered biotic interactions. This is different. It's when the plastic itself, either physically being there or maybe leaching chemicals, changes the local environment. And that change gives non-native species an advantage over the native ones already living there. The pollution actually helps the invaders compete.
So the plastic kind of selects for the tougher, maybe more opportunistic organisms, the invaders.
That seems to be the mechanism, yeah. The review points to studies showing these synergistic effects. Like one study found the invasive plant Solidago canadensis, when combined with microplastics in the soil, actually suppressed the root growth of native rice.
So a double whammy for the native species.
Exactly. And another one found an invasive plant, Sagneticola trilobata, was just way more tolerant to pollutants than the native plants around it. So in polluted areas, which often have plastic, it gets a serious competitive edge.
That's worrying. We're essentially creating habitats, polluted ones that are tailor-made for certain invaders. Okay, what about pathway three? This one sounded a bit different, more about the research process itself.
Yeah, pathway 3 is the experimental use of non-native species. This is less about a direct ecological interaction in the wild and more about a bias in how we study plastic toxicity. Basically, a lot of standard lab tests, the ecotoxicology assays looking at how harmful plastics are, often use non-native species as their test subjects.
Why would scientists choose invasive species for lab tests on plastic harm?
Well, often because they're known to be really robust, they're widespread, easy to find and collect, and, crucially, easy to keep alive and handle in a lab setting. There might already be established, standardized methods for working with them, so researchers might test nanoplastic effects on, say, the zebra mussel Dreissena polymorpha because it's a well-studied, tough invader.
Hold on. If we're testing how bad plastic is using species that are known invaders, species that are already super resilient and dominant,
Yeah.
aren't we kind of setting the safety bar too low? I mean, wouldn't native, maybe more sensitive species be way more affected?
That's exactly the concern raised. Using these really tough non-native organisms as our benchmark might lead us to systematically underestimate the real ecological risk of plastic pollution, especially in cleaner environments where native species haven't been pre-selected for toughness. It's a methodological convenience that could have real-world blind spots.
That actually leads perfectly into the next point. We've got these 3 interaction pathways, but the review also found the research itself is really fragmented and biased, right? Our picture's incomplete.
Very incomplete. The field's growing fast. Publications really took off after 2017, though there was a dip around 2021, maybe COVID-related. But yeah, it's super uneven. One of the key findings is this huge geographic bias, a massive asymmetry in where the research is actually getting done.
Okay, so where is it happening?
It's heavily concentrated in the global north. They see North America, Europe, Asia dominating. Specifically, the US, China, and Italy are publishing the most.
And why there?
It boils down to structural factors, really. Access to major research funding, having the expensive advanced analytical gear like the FTIR and Raman spectroscopy needed to identify tiny plastic particles, and just having established international science networks.
Right, the resources drive the research focus. But what about places where plastic pollution is maybe hitting hardest and invasive species are rampant, large parts of the world?
That's the worrying gap. Africa, Southeast Asia, Central America, Eastern Europe. They're severely underrepresented in this literature. It's not because the problem isn't happening there. It almost certainly is, maybe even more intensely. It reflects these structural inequalities, lack of funding, maybe language barriers, keeping local studies out of these big international reviews.
And if we don't have the data from those vulnerable regions, how can we possibly make effective global management plans?
We really can't. And it gets more complicated. Even in wealthy countries with good infrastructure, there can be biases, like
Yes.
Australia and New Zealand. They have great scientists and labs, but their conservation funding has historically been laser-focused on, say, terrestrial invasive vertebrates. Think rabbits, foxes, possums, big eradication programs.
Right.
So the whole issue of how those invaders might interact with plastic pollution just hasn't been a major research priority or funding target. It falls between the cracks.
Which leads us to taxonomic bias, too, really.
Yeah.
The review said the research focuses on a narrow slice of life.
Very narrow. It's overwhelmingly focused on aquatic environments, marine and freshwater. And within those, it's mainly aquatic invertebrates getting the attention, especially mollusks like snails and mussels and arthropods like crabs and insects.
Why them? Back to the lab convenience.
Partly, yeah. Familiar critters often used in those standardized tests we mentioned. But it means terrestrial ecosystems, which are obviously huge areas for plastic litter and potential dispersal routes, are critically understudied. And vertebrates get very little attention in this context, too. We could be missing a massive part of the picture by ignoring what's happening on land.
Okay, let's pivot to the plastic itself. Did the review find biases in terms of what size of plastic gets studied?
Oh, absolutely. A very strong size-based detection bias. Most of the studies looked at microplastics, those particles smaller than 5 millimeters.
Okay.
Macroplastics, the bigger stuff you can easily see, get some attention too. But nanoplastics, the really tiny ones, and weirdly mesoplastics, the sort of medium-sized bits, are rarely studied.
Why the obsession with microplastics?
Yes.
Just because they're everywhere.
It's partly that they are incredibly abundant, but it's also about the tools we have readily available. We've got established, validated methods, especially those spectroscopy techniques, FTIR and Raman, that work well for identifying particles in that microscale range.
So we study microplastics because we can see them relatively easily with current tech. But we might be overlooking the ecological impact of other sizes.
That's the risk.
Yeah.
Nanoplastics, for example, are potentially super important ecologically. They're small enough to cross biological membranes, get inside cells, but they are really hard to isolate from environmental samples, hard to detect, and you need even more specialized, expensive equipment.
Right.
And mesoplastics, sort of between 1 and 10 millimeters, they kind of fall into this analytical gap. Often too small for the methods used for large debris, but maybe too big or awkward for the standard microplastic analysis workflows that get missed.
You mentioned the tools FTIR and Raman. Can you quickly break down what they do in this context?
Sure. FTIR, that's Fourier Transform Infrared Spectroscopy. It's the most common tool reported in these studies. It basically works by shining infrared light on a sample and seeing how the molecules vibrate. Different plastics absorb light differently. It's good for identifying particles bigger than about 10 or 20 micrometers. It's used, for example, to confirm the type of polymer found inside non-native crayfish, like Procambrus clarkii, which are sometimes used as bioindicators in rivers.
Okay, and Raman is for even smaller stuff.
Exactly. Raman spectroscopy generally offers higher spatial resolution. It's better for identifying really small particles, sometimes even below 1 micrometer. It works on a different principle involving light scattering.
And basic microscopes are still in the mix.
Oh yeah, microscopy is still fundamental. It's versatile, relatively cheap for initial screening, just looking for potential plastic bits. Often it's used first, maybe combined with simpler validation methods like the hot needle test, poking a particle with a hot needle to see if it melts or curls like plastic before moving to spectroscopy for confirmation.
Makes sense. And what about the types of plastic polymers being found and studied? Is it mostly the common stuff, or are researchers finding more exotic materials interacting with species?
It's overwhelmingly the usual suspects. Which, isn't that surprising given what we throw away? Polyethylene PE, think plastic bags and bottles. Polystyrene PS, like styrofoam. And polypropylene PP, used in containers and textiles. Together, those three make up over 70% of the polymers reported in these interaction studies.
Which matches global production, right? Packaging, single-use items.
Exactly. Those are the plastics most likely to leak into the environment in huge quantities, so naturally they're the main ones acting as rafts and contaminants in these ecological scenarios.
Okay, this review clearly lays out the problem and the gaps. It feels like a roadmap for what needs to happen next. So what does this all mean for future research and actually managing this mess?
Well, the big call from the paper is for a shift towards mechanistic studies. We need to move beyond just finding correlations, like we found this plastic piece with this invasive species on it. We need research that establishes causality. How exactly did the plastic help the invasion? What was the mechanism? We need to quantify things like rafting dynamics, especially in freshwater, which is way understudied, and really figure out those aerial dispersal pathways. How significant are they?
Right. And once an invader is established in a plastic-polluted area, what then? How does the plastic change its impact?
That's another critical area, trophic transfer and bioaccumulation. We urgently need to investigate if dominant non-native species are acting as major pathways for plastics and the toxic chemicals often stuck to them to move up the food web. If an abundant invader eats a lot of microplastic and then gets eaten by fish, which are then eaten by birds or people, that dramatically increases the risk to top consumers, including us.
We talked a lot about the biases, geographic, taxonomic, size. How do we actually start fixing those?
It's a big challenge. The review argues strongly for more comprehensive comparative studies. We need work across different biomes, comparing terrestrial, freshwater, marine interactions. And crucially, we need that research happening in those underrepresented regions using standardized methods so we can compare results globally.
Which means?
It means serious international collaboration and really significant investment in building local scientific capacity, the labs, the equipment, the training, particularly in the global south.
Okay, and from a policy or management angle, how can regulators use this knowledge now?
The key message is integration. We absolutely have to start integrating plastic pollution concerns into non-native species risk assessments and vice versa. Management plans can't keep treating these as separate silos. The paper even suggests using predictive modeling, maybe even AI tools, to forecast where these two stressors are most likely to overlap and cause problems, so we can target interventions.
I have to say, the idea of using the invaders themselves to track the pollution was really interesting.
It is a neat inversion, isn't it? Some of these non-native species are consistently found in heavily plastic-polluted areas, and they seem quite tolerant. So they could potentially be used as cost-effective biomonitors. Their presence, or maybe the amount of plastic they accumulate, could give us a reading on local pollution levels, especially in places where doing high-tech water or sediment analysis is difficult or expensive.
It really hammers home the core takeaway from this review. Plastic pollution and biological invasions aren't just two separate crises happening at the same time. They're systemically intertwined, feeding off each other.
Absolutely. And the fact that the research fields themselves haven't been well integrated has really slowed down our ability to see the whole picture and develop truly effective, holistic strategies.
It costs ecosystems and us valuable time and resilience.
It really does.
Okay, so here's a final thought to leave you with. If plastic debris is fundamentally changing environments, acting as transport, as habitat, as a selective pressure, do we need to start thinking about heavily polluted areas differently? Like, should we consider things like the big ocean garbage patches or riverbanks choked with plastic as entirely new synthetic biogeographic regions, places where the old rules of invasion ecology might not even apply anymore? Something to think about until our next deep dive.